Good evening, everyone. Welcome to today's session on pre-formulation studies and BCS classification. I'm Mrs. P. Nagharita, Associate Professor from the Department of Pharmaceutics. And today's presentation will provide you with the insights into the foundational principles of dosage form design, mainly focusing on how pre-formulation studies and the BCS classification plays a vital role. So, in order to begin with, first in this particular webinar, we are going to discuss about what actually the uh, pre-formulation studies are and how this BCS classification it is going to have a significance in the pre-formulation studies. So let's begin. Pre-formulation studies, they form the early stage development of the drug. And also the main objective of this particular pre-formulation studies is to characterize the physicochemical properties of the drug candidate. So these studies, they always provide a crucial information for the successful formulation of stable and effective dosage form. On the other hand, we have the biopharmaceutical classification system, which we also call it as simply as BCS. It is a framework that classifies the drugs based upon their solubility and permeability. These two are the main parameters on which this BCS has classified the drugs into various classes. And this classification is mainly essential in order to know its role in the dosage form development, at the same time guiding the regulatory requirements during the drug development. And moving on, we have to see about the goals of pre-formulation studies. So here, the goals of pre-formulation studies, they are multifold. So to enhance the and understanding of the drug properties, and also to select suitable excipients which improves the drug performance, to optimize the drug formulation as well as stability, and to predict how the drug behaves inside the body. That is what we call it as a in vivo behavior of the drug. And also to support the regulatory documentation by providing detailed scientific data. So together, all these goals ensure the development of a safe and effective usage of the drug. Okay, so ultimately we need the effectiveness in the patient, and also it should be safe as long as the patient is being under that medication. So coming to the key components of the pre-formulation studies, it is mainly divided into three categories. The first one is the physicochemical properties of the drug, which includes the solubility, partition coefficient, ionization constant, and polymorphism. And then coming to the drug stability, it includes thermal stability, that is the temperature, then oxidative, photostability, and also compatibility with the excipients. And finally, we have the biopharmaceutical properties such as permeability, dissolution, and bioavailability. And each of these components, like either physicochemical properties or drug stability or even the biopharmaceutical properties, each of them plays a vital role in understanding the drug and designing its formulation. So moving on, like each of the, under each we have different subheadings, right? In physicochemical properties, we have solubility, partition coefficient, ionization. So in detail, we'll be seeing about each of the properties. So first coming to the solubility properties, we know that it is a critical parameter. The solubility is considered as a critical parameter in drug development because it directly impacts the drug absorption. Okay, when it is impacting the drug absorption, eventually it impacts even the bioavailability. So poorly soluble drugs often they face the challenges in achieving the therapeutic plasma concentrations, which limits their efficacy. And now let me break down this into subheadings like methods for determination, and then what are the factors that are affecting the solubility. So it have importance. The solubility have most important role in the drug absorption. Okay, because it plays a very crucial role in dissolution of the drug in the GIT. Okay, because this is where all the drugs get absorbed. So according to the Noyes Whitney equation, Okay, according to the Noyes-Whitney equation, the dissolution rate of a drug, it is proportional to its solubility. And for oral drugs, especially the higher solubility, it ensures a better absorption into the systemic circulation. And with what methods you can determine this particular solubility? We have two methods: shake flask method and pH solubility profile. So this shake flask method, it is a traditional method which involves a shaking of a known quantity of the drug in solvent until the equilibrium is reached. And then the concentration of the dissolved drug will be measured by suitable analytical methods. And pH solubility profile is nothing but it mainly assesses the solubility across a range of the pH levels, usually from pH 1.2 to 7.5. And to simulate different regions, why we are taking only 1.2 to 7.5? Because starting from the mouth till the intestine, these are the various pH ranges we will be having in the GIT. And what are the factors that are affecting the solubility? It includes pH, temperature, particle size, and polymorphism. So first, let us see how pH is influencing the solubility. So drugs exhibit this pH-dependent solubility. For instance, weak acids like ibuprofen, they are more soluble in the alkaline pH, whereas propranolol, which is a weak base, it will be soluble in the acidic pH. And then the temperature, typically the solubility increases with increase in temperature, but there exist certain exceptions like the drugs which exhibit less solubility when the temperature is being increased. You must have studied in critical solution temperature in physical pharmacy. The same goes good here. And then coming to the particle size. So reducing the particle size, it enhances the surface area so that it enhances solubility, which was discovered by, described by the Oswald equation. Okay. And also polymorphism. Polymorphism is nothing but existence of crystalline form in more than one form is called as a polymorphism. So different crystalline forms of a drug, they may have a varying solubilities. Like for example, amorphous forms generally dissolve faster than that of the crystalline forms, right? So this is how the solubility, how you can determine the solubility in pre-formulation stage, and also how it is affecting the various factors and with what factors it is being affected. Then followed by this, we have partition coefficient. So partition coefficient is nothing but it determines how well the drug can distribute between the lipid and aqueous phases, which is also important for predicting its permeability and distribution in the body. So this one, with the help of this, you can determine how well it can permeable through and also how well it can distribute within the body. So the partition coefficient, you can define it as a ratio of drug solubility in lipid as well as the aqueous phases, right? So we have two methods of determining this one: shake flask method and the HPLC based techniques. In the shake flask method, the drug will be dissolved in a two-phase system and its concentration in each phase is determined by using certain assay methods. Whereas in the HPLC based system, it is more faster and precise using the retention times. We will be, okay, you'll be studying in your final year about in analysis, what is the retention time and all. So here, based upon its retention time, it will be, we can estimate what is the partition coefficient. So what is its relevance here is, for example, a high partition coefficient indicates that the drug is lipophilic. Okay. High partition coefficient, drug is lipophilic in nature. So when the drug is lipophilic in nature, it is favorable for crossing the lipid membranes, whereas highly lipophilic drugs have poor solubility, right? And a low partition coefficient, it always have poor permeability, which may require the modifications like prodrug strategies or even the use of permeability enhancers. Okay. And then moving on, we have polymorphism. So as I said, it is the ability of a drug to exist in multiple crystalline forms, and these forms can have different physical as well as a chemical properties. And these different physical and chemical properties are they can significantly affect the drug's performance. So uh, here it shows the polymorphism, it shows impact on solubility, stability, as well as the bioavailability. So how it is showing the impact on solubility, right? So here the polymorphs with higher energy states, they are typically more soluble, whereas certain polymorphs, they may be more prone to the degradation, which impacts the formulation stability. And also solubility differences, they directly affect the amount of drug absorbed in the body. So all these things together, they are going to affect the drug performance. And how you can determine this polymorphism is by using X-ray diffraction, differential scanning calorimetry, and Fourier transform infrared spectroscopy. So this X-ray diffraction, it is going to tell whether your compound is amorphous or a crystalline form. And DSC, it detects the changes in the heat flow, which is associated with the polymorphic changes. And FTIR, it identifies the specific functional groups and their interactions in order to form as a polymorphic form. So moving on, we have pKa and ionization. So here pKa of a drug, it is a measure of ionization in a solution, which directly impacts its solubility as well as the permeability. So significance of ionization, it is having both on the solubility as well as the permeability. And its relationship with pH will be always determined by using the equation called as the Henderson-Hasselbalch equation, where there will be different equations for weak acids as well as the weak bases, right? And here, you can determine it by two methods, that is potentiometric titration method and spectrometry methods. So in potentiometric titration method, you measure the change in the pH as the drug is titrated with the acid or a base. And then in spectrophotometry, it monitors the absorbance changes in a drug solution at varying pH levels in order to estimate the pKa value. So with this, we have seen all the properties that are related to the physico-chemical properties of the drug. So if we go back, as we said, three components that were there, physico-chemical properties. So we have seen about the solubility, partition coefficient, ionization constant, and polymorphism. Now we are going for the drug stability. So coming to the stability studies, here the stability studies always ensures that the drug retains its safety, efficacy, and quality for all along the shelf life of the product, right? So what is the importance of this stability is, it always ensures the drug safety and efficacy over the time. And we have various types of the stability studies like thermal stability studies, where we use the differential scanning calorimetry and thermal gravimetric analysis to assess how the drug is responding to the heat. Whether in the presence of heat, whether the drug is undergoing degradation or it is not showing any deviation from its usual properties. That is, we are going to see by using these thermal properties. And then photostability, that means what is its sensitivity towards the light. So once you expose the drug to the UV light to test what is its degradation to the light. And then we have oxidative stability. So oxidative stability is nothing but you are going to assess the impact of oxygen exposure on the drug. And then compatibility studies with excipients. This is the most and the very, very, very important type of pre-formulation study that we do for any type of the drug that is being formulated into a certain dosage form, right? So here you need to know what is exactly happening when the drug is coming in contact with the excipient. So you need to see whether your drug is properly compatible with the excipient or not. If it is not compatible, what type of incompatibility you are seeing, whether it is a physical incompatibility or whether it is a chemical incompatibility. Okay, again, we cannot go for therapeutic incompatibility because you do not have the formulation yet. Okay, so you can see only whether it undergoes some physical incompatibility or chemical incompatibility. If so, then what is the remedy that you are going to do? Are you going to change the excipient, or are you going to change the concentration of the excipient? So all these parameters can be assessed once you do the drug excipient compatibility study. So this drug excipient compatibility studies can be studied by FTIR, Fourier transform infrared spectroscopy. So when you take the FTIR of pure drug, and also when you take the FTIR of drug that is mixed with the physical forms of the excipients, you should not see drastic changes, or you should not see major changes in the IR spectra of the functional groups of the drug. If you are seeing major difference, then it is considered as there is an incompatibility between the drug and the excipient. In that way, we use FTIR techniques in order to determine the drug excipient compatibility. And then coming to the BCS classification, here, the biopharmaceutical classification system, it was introduced by Amedon et al. In the year 1995. And its primary purpose is to predict a drug's in vivo performance based on its solubility, permeability, as well as dissolution characteristics. Okay, so solubility, permeability, and dissolution characteristics. And what is the main purpose here is to predict what is the in vivo drug performance and also to guide the regulatory requirements. So why this BCS matters? Why we have to go for this BCS classification or BCS type of the drug classification? Because it is a very valuable tool for the drug developers to identify the formulation challenges in a very early stage. And also it guides the regulatory decisions, including granting bio waivers. Bio waivers are nothing but because of their special characteristics, they need not be classed or they need not be classified into any of the type of the class of the drugs. We call them as the bio waivers. And it also helps to predict the behavior in the gastrointestinal tract, which helps for designing a better dosage form. So the drugs are classified into 4 categories, right? We have class 1, class 2, class 3, and class 4. So here, as I mentioned earlier, the classification is based on 2 parameters, that is solubility as well as the permeability. So now let me explain each of this class. In the class 1, there will be high solubility and high permeability. Okay, so these drugs, they dissolve very quickly and also they are well absorbed. They do not have any problem with either the solubility or permeability of the drug. You can take the example as metoprolol. Metoprolol belongs to the class 1 of the drug whose solubility and permeability are high. We do not have any bioavailability problems for these type of the drugs. And then coming to the class 2, they are low solubility and high permeability. So these drugs, they face the dissolution challenges, but they have good absorption once they are dissolved because their permeability is high, only the solubility is low. So till it undergoes dissolution, there will be a problem. But once it is dissolved, it will be having a better absorption. Okay, and then moving on here, the example here you can have it is a lapatinib. Okay, and then we have class 3 where there is a high solubility and low permeability. Okay, here we have high solubility as well as a low permeability. So these drugs, they dissolve readily, but they have limited absorption due to the low permeability. For example, we can have a called as a metformin. And then class 4 we have is a low solubility. Here it is called as a class 4, where it is having both low solubility as well as a low permeability. And these drugs have the most significant formulation as well as the absorption challenges. And example we can take it is take as the carbamazepine. Okay, so these are the various drugs that comes under each solubility or each type of the class of the drugs. So coming to the key definitions that we have to use here are high solubility, high permeability, and rapid dissolution. So to better understand this BCS classification, we need these definitions. So a drug is considered highly soluble if its highest dose strength dissolves in 250 ml or less of water across a pH range of 1 to 7.5. Okay, for 1 to 7.5, at that pH range, if it is highly soluble, where its highest dose strength is soluble, and if it is dissolving in 250 ml or less of that water, then we consider them as a high solubility. And then coming to the high permeability, so a drug is considered as highly permeable if the edge or more. 85% or more of the administered dose. Okay. And also coming to the rapid dissolution. So rapid dissolution here, a drug, it demonstrates a rapid dissolution if 85% or more of the drug, it dissolves within 30 minutes in pH buffers of 1.2, 4.5 as well as the 6.8 buffers. In this way, you can define various definitions like high solubility, high permeability, and the rapid dissolution. And then moving on, we have applications of this BCS in the dosage form development. Here, the BCS classification, they have several applications in the drug development as well as the regulations. The first and foremost one is, it simplifies the formulation strategy. For example, class 1 drugs, which are highly soluble and permeable, they require minimal formulation interventions. However, if you go for class 2, 3, and 4, some or the other way, they need some improvements or they need some adjustments in order to go for a better formulation of the drug. Because finally, what as a formulation scientist we require, that is the drug should have the 100% efficacy and it is showing a better therapeutic efficacy in a minimum dose required. Okay. And then it guides the bioequivalence study. So for drugs or for highly soluble and permeable drugs, in vivo bioequivalence studies may be waived off. Okay. So these bio waivers, they definitely save the time as well as the resources. So this is what we call it as a bio waivers for the class 1 drugs. And also it predicts the absorption challenges for class 2, 3, and 4 drugs. So this BCS, it mainly highlights the specific challenges for each class of the drugs, which enables the early formulation optimization, right? So for class 2 drugs, the focus should be on the solubility enhancement. For class 3, the strategies have to aim on improving the permeability, whereas for class 4, both solubility as well as the permeability has to be enhanced. And then moving on, we have the challenges and strategies in the BCS classification. So each BCS class, they present unique formulation challenges and they require specific strategies to overcome them. For example, the class 2, the solubility enhancement should be there. So how you are going to enhance the solubility? By use of nanoparticles, by using the salt forms, or even by using the solid dispersions to increase the solubility. And then like for example, you can also take as lipid-based formulations in order to enhance the solubility. Then class 3 we have where the permeability enhancement should be seen. So use of permeation enhancers or prodrug approaches, they can be used for improving the absorption. For example, designing the control release formulations for metformin, it increases the effective permeability. And then class 4, you have to go for the combined approaches of both class 2 as well as the class 3, because it addresses both the solid systems, such as you can use a nano carriers, or you can also use nano crystals like co-crystals. Okay. So for example, carbamazepine co-crystals, they improve both solubility as well as the permeability. So this is how the challenges can be answered, or the challenges can be addressed in the BCS classification, based on the BCS classification. Then coming to significance of this pre-formulation as well as the biopharmaceutical classification system, especially in the stability. So both this pre-formulation and biopharmaceutical classification system, they play a critical role in stability testing as well as the formulation development. So what is its role in stability testing is, the pre-formulation studies, they mainly identify the degradation pathways like hydrolysis or oxidation or even photodegradation. And also they help in designing the stable formulations. And stability studies should also ensure that drug meets their shelf life requirements. And then what is its role in, what is the contribution of the BCS is, the solubility related stability issues such as precipitation or crystallization can be better understood through this BCS classification. For example, like class 2 drugs with low solubility, they may experience instability because of the solubility limited dissolution in the formulations. And then coming to the analytical techniques in the pre-formulation studies, we have spectroscopic methods. So several analytical techniques are used to support the pre-formulation studies and also understand the properties of the drug candidates. The first one we have is the spectroscopic methods. In this, we have FTIR and UV visible spectroscopy. So this FTIR and UV visible spectroscopy, the FTIR mainly identifies the functional groups and drug excipient interactions. UV method we use to measure the drug concentration and solubility in the solutions, right? And NMR also is used for such conditions. And then thermal methods we have, like differential scanning calorimetry and thermal gravimetric analysis. This DSC, it assesses the thermal stability as well as the polymorphism, whereas TGA, it mainly monitors the changes in the weight due to the thermal degradation. And then the third category of the analytical methods we have is the chromatographic methods, where we use HPLC as well as the GC methods, which analyze the purity and stability and the drug compatibility with the excipients. And finally, we have for polymorphism, it is XRD, X-ray diffraction studies, which is used to study the crystalline nature of the drugs and they detect the polymorphism, right? And then moving on, we have regulatory aspects. So these pre-formulation studies and the biopharmaceutical classification system, they play a crucial role even in the regulatory submissions also, like IND and NDA, investigational new drug and new drug application. So how it is useful in IND and NDA submission is, the detailed data from these pre-formulation studies are essential for both of these submissions, IND as well as NDA, like solubility, stability, compatibility with the excipients, everything you have to include them in the dossiers. All the dossiers of these applications, they have the column where you have to give all the pre-formulation studies. And also, it helps in bio waivers for the class 1 drugs, like they eliminate the need for in vivo bioequivalent studies, which saves the time as well as the resources. And also this dissolution testing based on, so the dissolution testing and the BCS complaints, okay, it ensures with the regulatory standards which we receive it from the FDA as well as the ICH. Okay, so in short, if I want to summarize, okay, if we need to summarize, the pre-formulation studies, they act as a backbone Okay, so these pre-formulation studies, they form the backbone of the drug development by characterizing the drug's properties and also ensuring the stability and also helping in the formulation design. Whereas the BCS classification, it provides a framework for predicting the drug performance and optimizing the formulation strategies. So together, these both approaches, that is the pre-formulation and also the biopharmaceutical classification system, they simplify the drug development, they overcome the formulation challenges, and also ensures the regulatory compliance. And all these together, they lead to the successful development of the safe and effective dosage forms, right? So this is why we need to study about this pre-formulation and at the same time the BCS classification of the drug and how important it is in designing a very effective and a safe dosage form, right? So with this, we have seen all the properties that are related to the pre-formulation, stability, as well as the BCS classification of the drug. Hope you have understood well about this topic. If you have any doubts, please feel free to post your doubts in the comment section below. We would definitely answer to your questions very soon. Thank you so much for attending today's webinar.